Hello, everyone. This week we're going to talk about exercise 7, which is Introduction to Mendelian Genetics. So today I'm going to go over activity 1 and 2, and then I'll go over the rest of the activities tomorrow. I want this to kind of match up with your guys' lectures, so I don't want to overwhelm you guys with information and it just kind of go at the same pace. I also know you guys just had your second lecture exam, so I want to kind of hold off on uploading this information. Um, we will be having our first lab practical next week, and I will give you guys more information regarding the lab practical at the end of this week. But I highly suggest you guys start studying and start looking over the study guide that is provided in your lab manual. I'll post exactly what page it's on. I have an older lab manual, so for me it's on page 93, but I don't believe that's going to be the same for the new lab manual. Okay, so I'm just going to share my screen here. Okay, so now we're going to be talking about genetics and heredity. And heredity is this transmission of traits from one generation to the next. And genetics is this scientific study of heredity, how traits are going to be passed from one generation to the next, how these traits are essentially inherited. So the scientists that looked at this in great detail is Gregor Mendel. He is a botanist who studied this in pea plants, and he essentially chose pea plants because they reproduce very easily and he has a lot of control over their reproduction. And there's also a lot of there's a lot of characteristics you can look at when you study pea plants. You can look at plant or a flower color. You can look at pea shape. You can look at the or the seed shape, seed color, the pod color, pod shape. There's a lot of features that you can look at. So this is why he began to experiment with pea plants. So when he was doing these experiments, he didn't know about DNA or genes, but I'm still going to go ahead and define those because at that point he was just calling them traits, but genes are units of heredity, and these genes are sequences of DNA that are passed from parent to offspring. Genes control the characteristics an offspring will have. So as you can imagine, if you have two genes, one for your father and one for your mother for brown eyes, then you're more than likely going to have brown eyes. Um, if you have the gene for freckles, if you get the gene for freckles from both your father and mother, then you're more likely to have freckles. Things get a little different when you have when your father has blonde hair and your mother has brown hair. Then he was interested in knowing, would the offspring have a blended um, I guess, feature or trait, or would it be one or the other? And that was really what he was interested in studying when he did his experiments in pea plants. So I kind of want to go over the technique he did. So the first thing he did was obtain this pea generation or parental generation, and he had to essentially get these pure bred parents. So he had to make sure that this parent um, only produced purple flowers and this parent only produced white flowers, and he essentially would self-fertilize these um, for so many generations until he obtained this pure parental generation of purple flowers and this pure parental generation of white flowers. And then he crossbred these. So he crossbred the purple flower with the white flower, which led to the F1 generation. And when he crossbred these, well, the first thing is the way he ensured self-fertilization. So he self-fertilized this plant here, this to only generate purple flowers. And the way he ensured that is he essentially put a bag over it, or some kind of meshing over the purple flowers, so that the purple flower could only fertilize itself. So the way flowers fertilize themselves is that they have a stamen, which is shown here. These stamens um, are sperm producing pollen, and they also have a carpel, which is shown here. And when the pollen stimulates this carpel, that is how it is able to self-fertilize. So like I said, he would put a mesh over the purple flowers and a mesh over the white flowers so that they had to self-fertilize themselves. So once he obtained these pure plants, he crossbred them, and the way he crossbred them is that he would take off the stamens. So here it shows he took off the stamens of this purple producing or purple flower, and so it's only left with the carpel, which is shown here. And then he would grab a paintbrush, pick up the pollen from this white producing flower, and stimulate the carpel of this purple producing flower. And when he did that, he saw that all the plants had purple flowers. So in the F1 generation, all the plants had purple flowers. So first he saw, okay, when these organisms breed and produce offsprings, the offspring is not blended. It seems to have one unit versus the other. So either seems to be purple flowers or white flowers. In this case, it was purple flowers. So then he took this a step further, and then he bred these purple flowers. And when he did that, it led to the F2 generation. And in this F2 generation, he essentially saw that 75% of the plants had purple flowers and 25% of the plants had white flowers. So it was a 3-to-1 ratio. So he was kind of shocked to see these white flowers, and he realized that the F1 generation must have had the information to produce white colored flowers. However, it didn't produce it during this generation. So although all of these were purple flowers, they had to have that information from their parents so that we could see this white flower appear again in the F2 generation. And this just shows the cross breeding of the parental generation. So here, the purple flowers are shown over here, the parental generation. And there should really only be one flower shown here and one white flower shown up here. There should not be two. This is a mistake on whoever made this image. But there should be one purple flower here. And geneticists use these uppercase letters, which is this uppercase P, to represent dominant alleles, and I'll go over this in a bit. But essentially, in the P generation, the purple flower has two uppercase P's. And then in the parent generation, the white flower has two lowercase P's. And geneticists use these lowercase letters to represent recessive alleles. So when you cross these, you see that you get one uppercase P, one lowercase p, one uppercase P, one lowercase p, one uppercase P, one lowercase p, one uppercase P, and one lowercase p. And now the genotype for the F1 generation is going to be one uppercase P and one lowercase p for all of them. And since the uppercase P is the dominant allele, all of these are going to have purple flowers. So they carry the information of the white flowers as shown by the lowercase p. However, they don't actually express or these white flowers. So they don't have this appearance of white flowers. They only have the appearance of purple flowers. So Next, he took these F1 generations and bred those. So the F1 generation has the genotype of an uppercase P, lowercase p, and that is shown here. And he self-fertilized these. And here in the F1 generation, he self-fertilized these, and he saw when he self-fertilized the uppercase P with the lowercase p, uppercase P with the lowercase p, when you, I'll go over how to do these Punnett squares in more detail tomorrow, but when you cross these, you get one uppercase, I'm sorry, when you bring this down, you get a uppercase P, and then you bring this over here, you get an uppercase P, lowercase p. Bring this over here, uppercase P, lowercase p, lowercase p, lowercase p. And essentially what you see here is that you have 1/4 uppercase, 2 uppercase p's, 1/2 uppercase P, lowercase p, and 1/4 lowercase p. However, all of these that have the uppercase P are going to express the purple flowers, which are shown here. So whether it's 2 uppercase P's or uppercase lowercase p, uppercase lowercase p, they're all going to express these purple flowers because that is the dominant allele. And the only one that's going to express the white flowers is going to be this lowercase p, lowercase p genotype, which is shown here. So there's a 3-to-1 ratio. So Mendel also did this study and looked at other characteristics aside from flower color. He looked at flower position, seed color, seed shape, pod color, and stem length. And he always saw this 3-to-1 ratio where 75% of the pea plants would express one feature, the dominant feature, and 25% of the pea plants in the F2 generation would express another feature, the recessive feature. So just going back to what we know so far is, as we discussed previously, is that these are called chromosomes. These chromosomes are made up of DNA. So as you can see, the DNA wraps around these histoproteins so that it's nice and compact, makes our chromatin, and it gets packed in these chromosomes. These chromosomes, which is shown here, a single chromosome is shown here, can carry different genes across the chromosome. So the chromosome depicted here carries gene B, which can code for one thing. In this example, it codes for eye color, and then carries gene A, which could code for something else. And in this case, it codes for freckles. So if you remember, the human body cells has 46 chromosomes. 23 come from your mom, 23 from your dad. And a matching pair from mom and dad is called homologous chromosomes. So if we remember back to meiosis, our homologous chromosomes have to pair up in that first phase, or prophase 1. So a pair of homologous chromosomes, one from each parent, mom and dad, which is shown here. And this, when they're homologous, it essentially means that they are going to have the same genes in the same places along their chromosome. And the location of this gene is called a locus. So the locus for freckles would be here, and this is the location for the gene for freckles. So both chromosomes have the same genes, but can have different versions of these genes. So if you think of eye color, your mom might have blue eyes and your dad might have brown eyes. So then you have you carry the characteristic or the trait for both blue and brown eyes. So you can have different alleles. And this are different versions of these genes, and this is referred to as alleles. So you can carry the version for blue eyes and the version for brown eyes, and then So as we've mentioned, for each characteristic, inherited characteristic, an organism is going to inherit 2 alleles, one from each parent. So we already talked about you can inherit the allele for blue eyes and the allele from brown eyes. And those alleles may be the same or they may be different. So an organism that has 2 identical alleles is going to be homozygous for that gene. So here you have 2 identical alleles for the gene for freckles. They're both lowercase a. So that is an example of a homozygous gene or allele. An example of a heterozygous is when an individual inherits different alleles from both parents. So here, an example of a heterozygous chromosome would be the gene for eye color, which is uppercase B or brown eyes from dad, and lowercase b or blue eyes from mom. So you can either have homozygous alleles or heterozygous alleles, and so is the individual who has these chromosomes homozygous or heterozygous for freckles? So let's look at freckles. Is this individual going to be homozygous or heterozygous for freckles? They're going to be homozygous for freckles. They both have the same instructions or the same allele. They're both lowercase a's. Let's look at what about for the other gene seen here? So if we look at gene for eye color, you can see that they are going to be heterozygous for this allele. If you look at the gene for making a protein that is involved in controlling cells division, they're going to be homozygous for this allele. They are going to be homozygous for this R allele, lowercase r allele, and homozygous for this lowercase t allele. So if an individual is heterozygous, one of the alleles is going to determine the appearance of the organism. Okay, so let's, for example, you get a uppercase B from dad, which is for brown eyes, and a lowercase b from mom, which is for blue eyes. What is the offspring going to express? Well, the offspring is going to express whatever the dominant allele is. And like I said, geneticists, they use uppercase letters for dominant allele. So in this case, the dominant allele would be the brown eyes. So the offspring is going to, or the organism is going to express brown eyes. So the recessive allele, so if you are heterozygous for eye color, as shown here, the dominant allele is the uppercase B or brown eyes, and the recessive allele is going to be the lowercase b or blue eyes. So this allele has no noticeable effect on the organism's appearance, and like I said, it is geneticists show this recessive allele by lowercase letters. So as I said, brown eye color is dominant to blue, so uppercase B, lowercase b would equal brown eyes. So the genotype for this individual in terms of eye color would be the uppercase B, lowercase b. This is the description of the allele combination. The phenotype is going to be the physical outcome of this genotype. So in this example, the phenotype would be brown eyes. This organism is going to have brown eye color in the end. So you can also use these words. So you can also be like a homozygous dominant. An example of so if you had to see, like if you were asked which allele pairs are homozygous dominant in this example, you would say that these capital G's here are going to be homozygous dominant. If you were asked which ones are going to be homozygous recessive, then you would say here the gene for freckles is homozygous recessive. And then if you were asked which allele pairs are heterozygous, you would say that the gene for eye color, the uppercase B, lowercase b, are heterozygous alleles, or these alleles are heterozygous. So the other thing that Mendel observed when he did these experiments is the law of segregation. And essentially, a sperm, which is shown here, and an egg is going to carry only one allele for each inherited character. So here the sperm is going to carry the allele for purple flowers, and the egg is going to carry the allele for white flowers. And at fertilization, the sperm and egg are going to unite and contribute its alleles to the offspring. So the offspring is going to have two alleles. It's going to have this uppercase P from the sperm and this lowercase p from the egg. So this offspring is uppercase P, lowercase p. So if we go back to our original example of the plants, the pea plants with the purple flowers, the F1 generation had this genotype, the uppercase P, lowercase p. Well, when these purple flowers self-fertilize, their alleles separated during the production of gametes. And remember, we had the uppercase P, lowercase p on the top. And I should go back over here. So like I said, they separated during the production of gametes, these uppercase, lowercase p plants. And here you can see that you have the half p, half lowercase p, half uppercase p, half lowercase p. So that is the law of segregation. When sperms and eggs unite at fertilization, each contributes its alleles, restoring the paired condition in the offspring. And then we also have the law of independent assortment. And here, we already kind of talked about independent assortment when we learned about meiosis. If you recall there, it's essentially that these alleles are going to separate independently of one another. And this is especially true when they're on different chromosomes. So these traits are not inherited as a package deal. So for example, Mendel studied this in different pea plant characteristics. So he looked at seed shape. So round is dominant when it comes to seed shape, and he looked at seed color. So yellow is dominant when it comes to seed color. So for seed shape, round is dominant, wrinkled is recessive. When it comes to seed color, yellow is dominant and green is recessive. So he basically tracked these characteristics one at a time using monohybrid crosses, and he was able to see that, well, one, he was able to see that the trait for seed shape, the round trait for seed shape, is dominant compared to the wrinkled, which is recessive, and that the yellow is dominant compared to the green, which is recessive. And then he also did dihybrid crosses, which I actually want to talk about this more when we talk about crosses tomorrow. But so yeah, I'll probably talk about this more tomorrow, but just very briefly, essentially what he did is when he crossed these, he saw that individuals could produce 4 possible gametes. Like you can have the round yellow pea. You can have a wrinkled yellow pea. So round yellow pea, wrinkled yellow pea, a round green pea, and a wrinkled green pea. So the way the R and the way the capital R and lowercase r segregate does not influence the way the uppercase y, lowercase y segregate. So all of these combinations are possible, and we'll go over this more tomorrow. But in activity 1, you will, so in activity 1, we would have done this as a class, and you would look at various dominant and recessive traits in the classroom population and see how they are, see what kind of phenotype, genotype, or phenotype is in the classroom. So we would look at widow's peak, unattached earlobes, freckles, and brown eye color. So the first thing is you have to document your phenotype. So what is your phenotype? Do you have a widow's peak? So in my case, I do not. Do you have unattached earlobes? In my case, I do. So that's a yes. Do I have freckles? I have freckles. And is my eye color brown? Yes, it is. So the only thing I don't have is the widow's peak. And then you record your phenotype for these traits in the table on your lab manual. I believe this is the old page number. I didn't update that. And if you have the dominant trait, write a capital D for dominant in the first column. So you would document your phenotype here. So like I said, I'd be a capital D for freckles, or for unattached earlobe, freckles, and eye color, and I'd be a lowercase d for widow's peak. And then if we were in a class with 30 students and counted the number of students with dominant traits, we'd probably see something like this. So 14 people would have this dominant phenotype, 16 would have the recessive phenotype, meaning they don't have a widow's peak. So, and then if we looked at something like freckles, you would see that 6 people have this dominant phenotype while 24 have the recessive phenotype of no freckles. And for brown eye color, we would probably see about 26 people with this dominant phenotype and 4 people with the recessive phenotype. So these 4 people would have like green or blue eyes or something else. So are these dominant traits always more common? So are dominant traits always more common? As you can see here, some dominant traits are more common, such as brown eye color. However, some dominant traits, such as freckles, are not more common, as you can see by this 20%. Activity 2 would be you would look at homologous chromosomes, and in your lab manual, you should see a homologous chromosome as shown here. And you will use this homologous chromosome to answer the following questions. So which allele pairs are homozygous dominant? Remember, homozygous means that they have the same allele, and dominant is that they are the capital letters. So in this case, capital B is an example of homozygous dominant. So you'd go through and identify any other homozygous dominant pairs. Which allele pairs are homozygous recessive? So in this example, the lowercase h is an example of homozygous recessive. So you go through this pair of chromosomes and see if you could identify any other allele pairs that are homozygous recessive. Which allele pairs are heterozygous? So an example of a heterozygous allele pair would be this capital F, lowercase f, and you'd go through this pair of chromosomes and identify any other allele pairs that are heterozygous. So I'll go over activity 3, 4, and 5 tomorrow, and I'll make that available